Assalamu alaikum and good day. First of all, I would like to thank you for allowing me to present my study with the title, Simple Statistical Method for Determining the Ratio of Platelet to RBC. The platelet to red blood cell, RBC, ratio can be relevant in certain medical contexts, particularly in diagnosing and managing certain conditions like thrombocytosis, high platelet count, or thrombocytopenia, low platelet count, which can affect the blood's ability to clot properly. It can be assessed as part of a comprehensive blood analysis and provides valuable information on the circulatory system. Normally, the ratio between platelet and RBC in the blood is about 120 or 5%, which means more RBC compared to platelet. However, this ratio can vary depending on various factors including age, sex, and individual health conditions. It is important to note that interpreting the platelet to RBC ratio requires consideration of various other factors, including the absolute counts of them. Therefore, this study provides a basic cost-free method for blood cell count that may enhance the knowledge and skills of healthcare students, especially those involved in medical laboratory activities. Blood cell counting remains an essential skill for medical laboratory students, particularly in teaching and learning settings with resource-limited environments. In this study, the ratio of platelet to RBC can be indirectly assessed by examining the complete blood count, CBC. The number of platelets and RBCs is counted within the defined area known as the volume of examined blood on the smear image. This count is typically expressed as the number of blood cells per unit volume. To ensure accuracy and reliability, manual blood cell counts are often performed in duplicate and triplicate to compare the consistency of counting. The ratio can then be calculated by dividing the platelet count by the RBC count. Analyzing blood smear images can provide valuable information concerning the distribution and morphology of blood cells. Sometimes basic statistics tools play a significant role in teaching, especially when there is a lack of resources for learning. Manual blood cell counting is a labor-intensive process and is usually reserved for situations where automated methods are not available or when there is a need to confirm results obtained from automated analyzers. The selected smear image for blood cell count has an average ratio of platelet to RBC 5.47% for the 6 different counts of blood cells inside the same defined volume as presented in table. The finding ratio value is almost near the actual value, which indicates the applicability of this simple method in teaching purposes. The result of this study provides the biostatistics tool and technique necessary to analyze data related to platelet to RBC ratio. It can be employed to analyze the relationship between these two variables and summarize the distribution of platelet counts, RBC counts, and their ratio in a given population. On the other hand, it may have many challenges as blood cells can vary in size and shape, which can affect counting accuracy. Irregularly shaped cells, such as sickle cells or fragmented cells, may also be miscounted. This proposed alternative method as a basic biostatistical tool enables healthcare students to gain valuable knowledge regarding blood count and at the same time to help them to understand blood features and functions.
Few articles were published regarding this work and it was recognized with many award. Thank you and have a great day ahead.